So our topic for today is gibberellins. Now this is a hormone or a plant growth regulator. Which plays a part in stem elongation and seed germination. Now let's begin with stem elongation. In this topic we're going to cover two things. One is tall plants and the other one are dwarf plants. Now tall plants have the dominant form of an allele called Le. So it can be homozygous dominant for this allele or even a heterozygous. Now if this dominant Le allele is present, it will code for a series of enzymes. In a pathway and the last enzyme in this pathway will code for an active form of gibberellin, and this plays a role in cell division, or it triggers cell division in stems and cell elongation. Both of this cause a plant to grow tall. In case of dwarf plants, a mutation occurs and the plant is homozygous, recessive for the allele small l small e, that is LE. Now in presence of this allele or this genotype, a substitution occurs in that last enzyme of the pathway. That is, thiorein is changed to alanine. Hence, the primary structure of the enzyme changes. So GA1 is not produced. That's about all we need to know for tall plants and short plants. That is the role of gibberellin in stem elongation. Now let's move on to seed germination. So let me draw this out. This is a rough sketch of a seed. Let this be the seed coat. Inside we have our embryo. The aleurone layer. And the endosperm. Let me label this out. Testa or seed coat, aleurone layer, endosperm, which contains nutrients, and our embryo. Now, in presence of water, the embryo secretes gibberellin. This gibberellin diffuses to our aleurone layer, where it stimulates a series of activities. We're going to elaborate on that now. In aleurone cells, we have a molecule known as PIF, phytochrome interacting protein, and we have another protein called a DELA protein. I'm going to elaborate on this soon. The main function of aleurone cells is to produce amylase, which is an enzyme, and this enzyme is coded for by a gene. So to transcribe this gene, we need PIF to bind to the promoter region. Let me clear this out, sorry. Promoter region of our DNA. So once the PIF binds to the promoter region of our DNA, this is going to transcribe the DNA and eventually code for our amylase. But this DELA protein binds to PIF and thus restricts it from binding to the promoter region of our DNA. So in the presence of water, this embryo produces gibberellin, which diffuses to our aleurone layer. And in this aleurone layer, let this be our gibberellin. Gibberellin. The gibberellin binds to an enzyme which causes this enzyme to be activated, and this enzyme in turn breaks down our DELA protein. And once this DELA protein is destroyed, PIF is free to bind to a promoter region and thus transcribe this DNA, which will in turn code for amylase. Now once the amylase is produced, this amylase diffuses out of the aleurone layer and into the endosperm. Here, starch is broken down to maltose with the help of amylase. This maltose will in turn be broken down to glucose, which will be used in respiration. And that's how the seed obtains energy to germinate. This glucose can also be converted to proteins and other nutrients which the seed might need to germinate. So that's about it for the whole action of gibberellin in stem elongation and seed germination. Hope you guys understood the concepts, and if there is any way I can improve my explanations, which I know there is a lot of scope for that, please note them down in the comments. And yeah, see you next time.